So after making purines and pyrimidine, now we have to look of the salvage pathway, or how these purines and pyrimidines are effectively used in the DNA synthesis, and then after usage they are eliminated from the body. So this is the purine salvage pathway. First, we have known that ribose 5-phosphate is converted into phosphoribosyl pyrophosphate by an enzyme, the synthase, PRPP synthase, and it is converted into inosine monophosphate, as we have previously discussed. IMP is dehydrogenase converted into GMP, or either it is converted into adenosine monophosphate. This process is inhibited by 5-mercaptopurine or azathioprine, and dehydrogenase is inhibited by ribavirin and mycophenolate. So IMP is either converted into here or here. This is converted into this. So now, how IMP is further converted into inosine and then hypoxanthine. IMP is converted into inosine, and inosine is further converted into hypoxanthine. Hypoxanthine is oxidized by xanthine oxidase, and it is converted into xanthine, and this xanthine oxidase is converted into uric acid, and this uric acid is oxidized urate oxidase, or we can use resburicase enzyme to convert this into allantoin, which is secreted into the urine. This is the final product. So here we have seen that IMP is converted into inosine is converted into hypoxanthine, xanthine, and uric acid, and then allantoin. These are the main general sketch of this conversion. The next thing is we can use hypoxanthine and a base which is guanine. This is monophosphate. This is a nucleotide. This is also a nucleotide. This is guanine. This is a base. So over here we also have adenine. So now we have to convert. We can use this synthase to make IMP. So now everything is made and used and secreted into the urine. So purine salvage pathway is it can convert the remaining guanine and adenine bases to further make GMP, IMP, and they reuse it one and after other. So how they reuse it? They can use an enzyme known as transferase, because they need to transfer again into nucleotides. So over here we also use transferase enzyme in order to convert back these into. So over here we have hypoxanthine and guanine. So the enzyme, and we also used over here is PRPP. So adding this PRPP with this enzyme, now we can convert hypoxanthine and guanine by this PRPP transferase enzyme back to the IMP and guanosine monophosphate by this enzyme transferase. And on the other hand, we also use PRPP over here to make this adenine converted back into adenosine monophosphate. So this is generally the whole sketch of how these things are work. Over here, this adenosine monophosphate is converted into adenosine. Since it's a adenosine, so it's a nucleoside. Over here we also have guanosine. So GMP, we remove the phosphate, it is converted into guanosine, which is a nucleoside. We remove the ribose sugar, so it's a guanine. Also adenosine, adenine monophosphate, which is a nucleotide, is converted into nucleoside, and then it is converted back into inosine by an enzyme known as deaminase enzyme or adenine deaminase, ADA. This enzyme is inhibited by a medication known as cladribine or pentostatin. They inhibit this conversion and inhibit the make of inosine, which converted into uric acid and further enzymes. So this is one way these medication affect their side effects on the body. And also we have this HGPRP enzyme, which is converted into, so if there is a deficiency of HGPRT enzyme, then we have the condition known as Lesch-Nyhan Syndrome, which is a deficiency of HGPRT, which is hypoxanthine guanine phosphoriboside transferase enzyme. If this enzyme is deficient, then we cannot salvage these purines. So we keep on making these purine and keep on making xanthine, hypoxanthine, uric acid, and it exerts its side effects. Also, if we deficient this enzyme, then it can cause a condition known as severe combined immunodeficiency, and how it will affect the human body we'll discuss here. The first thing is Lesch-Nyhan Syndrome, which is the X-linked recessive deficiency of hypoxanthine guanine phosphoriboside transferase. So it means no salvage, only synthesis going on. Whose synthesis? Purine synthesis. So there is purine synthesis increased, so we can increase making this inosine, hypoxanthine, xanthine, and uric acid, which is also called hyperuricemia, which can exert effect in as gout. And we have a pace-off situation in which there is aggressiveness and self-mutilation. We have the crystals, the red, orange crystals in the diaper, and we have a tense muscles known as dystonia condition. So the child, it is X-linked recessive, so it means it's a child condition with this deficient enzyme. no salvage, increased purine, increased uric acid, which can cause different type of problems in the body. So we have increased purine. We can also have macrocytosis, but this is very less common symptom. Self-mutilation, the patient is eating his own finger, nails, and there is a scar marks of it. And the treatment of choice for this condition is we symptomatically treat. First, we give allopurinol, and if it is not working, we can give fiboxate. These allopurinol, they inhibit these enzymes. Allopurinol inhibits xanthine oxidase, which converts hypoxanthine into xanthine, and fiboxate inhibit xanthine into uric acid. So we can limit this by these two enzymes. The next condition is, this is Lesch-Nyhan Syndrome. Next condition is adenosine deaminase enzyme deficiency. If this enzyme is not there, we can make adenosine, but we cannot make inosine. So the AMP is increased in the body. So if deaminase is not present, we know degradation of adenosine to inosine. So we increase the level of AMP, which is nucleotide, which is converted into increased deoxyadenosine monophosphates. And this deoxymonophosphate, generally physiologically, it feedback inhibit the reductase enzyme, which is converting the dihydro monophosphate into dihydro triphosphate. So the reductase enzyme is actually ribonucleotide reductase. It convert the UDP, redirect inhibition of ribonucleotide reductase. Generally it do this, but if there is increase in the level of the deoxyadenosine monophosphate, then it increase the inhibition of ribonucleotide reductase. So we cannot make making of UDP to deoxyuridine diphosphate, which is a precursor molecule of DNA or purines, pyrimidines. So there is no precursor molecule of DNA, no DNA precursors, so DNA is not formed. So the cell which use that DNA is lymphocyte. So there is no lymphocytes, and if there is no lymphocyte, then the patient is immunodeficient, and it is one of the cause of severe combined immunodeficiency. So this is the general mechanism of adenine deaminase deficiency. Also, if the patient is on cladribine or pentostatin, then also it can show the effect of reversible cause of severe combined immunodeficiency. And if we remove these two medication, then we can revert that condition. And this is all about the purine synthesis, pyrimidine synthesis, and their salvage pathways.